Now, prokaryotes are having two different domains, or we can say two different types. One is known as archaea and another is known as bacteria. Bacteria is also a eubacteria that we call true bacteria and cyanobacteria. So there is a two different types of bacteria. Simply we can say eubacteria, which is known as a true bacteria, and the another type of bacteria is there that is known as cyanobacteria. Now the second domain in prokaryote is archaea. This archaea is having different types of conditions. They face different types of conditions like extreme environments they face. For example, they grow very well in a deep sea, which is having a different types of hydrothermal vents. Then they also grow very well in a extreme salinity. So these are the archaea and the bacteria. Now, when we talk about the prokaryotic cell wall, simply we can call it as a bacterial cell wall because the best example of prokaryote is bacteria. So, bacterial cell wall, there is a cell membrane. There is no any organelle. So, it is easy to maintain cell shape, and the peptidoglycan, which is most important cell wall containing or cell wall supporting substance, peptidoglycan we can call it as it is a semi-rigid. Then it is made up of N-acetyl glucosamine and N-acetylmuramic acid. So this linking of acetyl glucosamine and acetylmuramic acid forms a peptidoglycan, and they are very simple, and it is present in almost all bacterial cell. Now there is a two types of bacterial cell. One is known as gram positive bacteria and gram negative bacteria. This peptidoglycan layer which is present in gram positive bacteria is very thick, and there is a teichoic acid, and due to that they shows this gram staining like a blue or violet color when we apply the gram staining method to the bacteria. Gram positive bacteria looks like a blue or violet color, whereas another bacteria is also there that is known as gram negative bacteria. In gram negative bacteria, the peptidoglycan layer is very thin. The outer membrane is periplasmic spaces are there, and due to these periplasmic spaces, we can say that there is no inclusion of the blue or violet color, and hence they shows the attachment of stain known as safranin, and due to that it will shows the pink color. So gram positive bacteria, when we observe under microscope, it shows purple to violet color, whereas gram negative bacteria, when we observe under microscope, it shows the pink color. Now moving to the next parameter in a prokaryotic cell, that is plasma membrane. Plasma membrane is a selectively permeable membrane, and it is present produces boundary between cell and the outer environment. It is made up of phospholipids and proteins. So there is a layer of phospholipids, and along with that phospholipid there is a presence of proteins. So phospholipid bilayer in a fluid state, it is nothing but the cell membrane present in prokaryotes. Bacteria have no sterols. There is absence of sterol except mycoplasma. No cell wall have cholesterol, and it adds no cholesterol to add rigidity. Now when we talk about the internal structure of prokaryotes, there is a presence of cytoplasm. This cytoplasm is semi-fluid, and there is also presence of ribosome. This ribosome is known as 70S ribosome and 80S ribosome. Then there is a presence of nucleoid region. One large circular chromosome or extra circle of DNA plasmid is also present in the prokaryotic cell. There is endospores production. Say for example in Bacillus and Clostridium species, they produce a endospore. These endospores are useful in a survival of the organism. Now when we talk about the external structure of prokaryotic cell, as we know that they are not very much developed, they are undeveloped. So we can say that there is very small amount of external structures are also there, like internal structure. Simply they show the presence of flagella. In some bacteria, in some prokaryotes, this flagella is also absent. But this flagella is useful for the motility of the microorganism, that is bacterial motility. Then there is a different types of flagella is also there. Say for example, peritrichus, monotrichus, amphitrichus. These are the types of flagella which is present on the body of the bacteria. Along with the flagella, there is a presence of pili, which is useful for the different parameter of like attachment of bacteria on the surface, and it also helps in a transfer of DNA between bacteria of two different sex with conjugation method. Now there is also presence of glycocalyx, which is a very small thin layer which is present outside to the bacterial cell. Along with that, in some bacteria, capsule, which is a very thick preventive structure present, is also present, and in some bacteria the slime layer is also present. Now here it is nothing but the simple structure of bacteria. It is also show the internal and external structure of bacteria. So at the external side you can see the capsule is present, which is very exterior part of the cell. After that there is a cell wall. This cell wall is protected by capsule, and after cell wall there is cytoplasmic membrane. Now coming to the next, that is there is a presence of flagella. A long flagella is there, which is a single flagella shown in this particular diagram. Along with the flagella there is a presence of pili over throughout the bacterial cell, and inside to the bacterial cell you can observe the nucleoid. That nucleoid is nuclear material which is present without nuclear membrane, and that's why it is known as nucleoid. Along with that nucleoid, in some bacterial cell you can or we can observe the plasmid, which is a circular DNA, extra chromosomal structure. We can say that it is present in prokaryotic cell. So this is nothing but the simple structure of prokaryotic cell. In next lecture we are going to concentrate on eukaryotic cell. So